The second speaker is me. So I'm a soil microbiologist. Soil microbiology, you know, we hear about the biologics and things like that. So soil microbiology has been attracting lots of attention lately. So I thought that you may be interested in knowing more about what or how we can use soil microorganisms to intensify agriculture in a sustainable way. I must first say that I would be biased. My interest is in improving the efficiency of nutrients, in particular nitrogen and phosphorus. So I will be talking with this bias. Before I talk about what we can do with microorganisms, I would like to talk a bit about how soil function to have kind of a refresher, and after that we can understand better. So the soil, as we know, is a living entity, and it is alive through the microorganisms that live there, but also because of the roots, and very importantly because of the roots. Here we have prairie plants, tall prairie plants with the root system. The soil is meant to contain roots. And this is wheat with lots of roots also. And if we could see more closely, as we're going to do very soon, we would see that roots do not exist alone. They are always in association with lots of microorganisms. What we see here is the mycelium of mycorrhizal fungi associated with that root here. And this mycelium is taking up nutrients, and all the nutrients in kind of available form for the plants. And these nutrients can be passed on to the plant roots through connection that the fungi do, with the roots. And in fact, that fungus, the mycorrhizal fungi, grow inside of the roots, as we can see here in a close-up. I have here a drawing which is maybe more easy to understand. So we can see the nutrients taken up by the mycelium of the mycorrhizal fungi, transferred to the plant through the connections that we call arbuscules. And this is a service that the mycorrhizal fungi give to the plant in return for carbon. Carbon is a very important currency in soil. Most soil organisms need carbon for growth and for deriving energy. And this carbon is coming essentially from plants. So plants are very important, and they support almost all microorganisms in the soil. And the root zone is a hotspot of microorganisms where we find lots of diversity and lot of function as well, because the plant roots feed the microorganisms. So this is very important to realize. The plant feed the soil microorganisms. So most soil microorganisms, if I can say, care for plants. Most soil microorganisms are going to be beneficial. Something also that is very important and that we have seen this morning is that the plants are not passive actors in this soil environment. They actively select the microorganisms that are going to be living in their surrounding. Plants have the ability to produce lots of phytochemicals, and with these phytochemicals, they can organize their biological environment. And we have example of this, very well-known example. Genistein is a compound produced by soybean that will turn on the genes necessary for the formation of nodule in the symbiotic bacterium Bradyrhizobium japonicum. So this phytochemical will induce the formation of a nodule that will culminate in the fixation of nitrogen for the soybean within root nodules that we can see here. Oops, sorry. Oops. Yeah, root nodules here where the bacteria will be living soon. Of course, we also have pathogenic microorganisms, but plants can defend themselves against pathogen. We have here a molecule of jasmonate. We've heard about that this morning too. It's a very well-known example. This molecule is produced by the plant under attack, and it turns on the mechanisms of plant defense. So we have the beneficial microorganisms, we have the pathogens. We also have microorganisms that are living in the root without causing any symptom, and sometimes without us knowing what exactly they're doing. In fact, if we could see through the roots, we would see that roots usually contain a lot of microorganisms. Some of them have an impact on plant growth. If we want to improve the efficiency of nutrient use, there can be many ways. I'm a microbiologist, so I think that maybe microorganisms could be managed to improve the efficiency of nutrient by plant because they have a large effect on plants. We can think of different ways through which microbe could be managed. We can think of using these signal molecules, maybe just plainly produced commercially. We can also think of using the plant themselves, maybe through genetic selection. We could select plants that can, naturally associate with more beneficial microorganisms. We have also crop rotation that is used since a long, long time. That is another way through which we do manage soil microorganisms. We can also think of isolating microorganisms and formulating into inoculants. So, of course, we would use the beneficial ones, and we could apply them onto seeds or to the soil. We can think also it might be possible to select microorganisms in the same way that we select plants. We can also think of engineering plants by putting in plant genome some genes that could give them, some genes from microorganisms, of course, genes that could give them additional pathways through which they could use soil nutrient more efficiently. So there are beneficial plant root associates, and we can isolate them, formulate them. There are, in fact, many soil microbes that promote plant growth, many microorganisms. They have different modes of action that can be classified into three main classes: hormonal, biocontrol of pathogen, and nutrient mobilization. And when it makes sense, that nutrient mobilization can be very good for improving the efficiency of nutrient, we also have to remember that hormonal and biocontrol are also going to be important to have better nutrient use by plants. Because if we have, for example, a plant that grows bigger and produce more yield because of hormonal effect or because it's more healthy, this plant and this crop will use soil nutrient better. So here we have an example on the hormonal effect. In this example, we have the bacterium BPUC26 inducing root branching. We can see the bacteria here. So it's producing a volatile compound that induces branching in here, whereas in the control we don't have branching. So modifying the morphology of the plant can be interesting to improve nutrient use. there was also a lot of interest in the associative nitrogen-fixing bacteria. And I have a list of the bacteria that were studied and are still under study. And why it is interesting is because nitrogen is such an important nutrient for crops. It's required by plants, of course, and it's required in very large amount. And nitrogen fixation brings in free nitrogen, which is very interesting. However, something to keep in mind is that associative nitrogen fixation, or even nitrogen fixation by endophytes, is not going to be as efficient as what happened with the rhizobium system. And the reason is that in the case of legume and the rhizobium, the bacterial associate, we have this structure that is the nodule that provides the ideal condition for nitrogen fixation to take place. Of course, probably we can improve the system with associative nitrogen fixation, but for now, most of the inoculants that are used are rhizobial inoculant for nitrogen-fixing legumes. We have seen also numbers earlier today on this. The second in importance are the phosphorus solubilizer, and then we have marginal utilization of other inoculants. However, there is increasing interest in inoculants. So we have to work more on this, and why there is less interest, well, less use of the inoculant other than rhizobial inoculant. It is because these inoculants are less reliable. So we have to understand that in the rhizobial system, the organisms are specific. So soybeans, for example, is nodulated by Bradyrhizobium japonicum. So the competition of Bradyrhizobium with other microorganisms in the soil is less important in this case. Also, the bacteria are in the root nodules, so they are somewhat protected from the variation in environmental conditions, which is not the case with the organisms that are associative. So we need to do more work to understand the soil system. And it is very, very complex, and actually that's why we know little at this point. Fortunately, we do now have a very powerful tool to study the diversity of the soil and the interaction between microorganisms and plants. And I would like to just take a minute to explain to you a bit how this works so you can understand the field. So high-throughput sequencing really is giving us a huge opportunity to improve our understanding of the soil function. And this is how it works. So we can take a soil sample these days, say rhizosphere soil samples, and we extract the nucleic acid. We sequence this nucleic acid with now this very powerful machine that gives us huge amount of data because the diversity of the soil is huge. So of course we have huge data if we want to understand what's going on. And then we can analyze this data. And we do that by comparison of the sequence that we get. So for example, if we get sequence of Glomus mossae, we can look into data banks that are built by people and maintained by very nice people. And we can see, okay, this sequence looks like Glomus mossae. So most likely, we have Glomus mossae in our soil sample. Similarly, we can look at the different sequence that we get, and here we have a sequence that is similar to a sequence of a genes coding for amylase, for example. So we have potential for amylase function in our soil. And even when the sequence do not correspond perfectly, because the organisms that are similar have similar function and are taxonomically related, we can classify them. So here I would like to point out that the progress that we are doing right now in the field of soil microbiology is due not only to the technology advance, in sequencing, but also because of the work of a lot of people that have created this data bank, and all the people that put data in this data bank, and also the very nice maths guys that develop software for us to be able to make sense of this huge data. So maybe I can point out just a few features of these things. Gene bank is very well known. It contains a lot of information on a lot of genes of a lot of organisms. The IMG data bank contains the sequence information of 30,000 sequence genomes, so this is a lot. Silva, Unite, and RDP contain information on specific genes that contain a lot of genetic information for the taxonomic classification of microorganisms. And we have software that can make this comparison of sequence. We have software that can classify the taxonomic data on fungi into functional group. We have the same thing here for the bacteria. We can infer function of bacterial community based on taxonomic information. And we have software that can take all these small sequences we get and tell us they are coding for which genes that is responsible for which function. And we have also the R project that is built by a lot of statisticians that give us the tool to analyze statistically the data and to create a very nice graph that actually makes sense of the huge data that we have. So what do we do with this? Well, we can know what's in the soil. For example, we can see which microbial species is consistently associated with a plant. So we can put effort on these microorganisms. We can also look at consortia that would be more beneficial for plant growth and consistently present with a plant, because that would resolve the problem of reliability that we have at the moment. Whoops. So we have here an example of the complexity of the data that we get. Here in this sphere, we have all these points are dominant bacteria in the soil. It's from an experiment looking at the effect of different type of fertilization on bacteria. So we can see that these bacteria could be grouped into network based on how they react with the environment, and then the authors could do some relationship between, the bacteria that form the different modules. So they got here patterns, and they could compare the patterns of the different treatments. My point here is that I want to show that we need more than microbiologists to make sense of soil diversity. And in fact, in this study, to get to this result, we needed to have the contribution of different disciplines to design the experiment and to interpret the results. So agronomy was required, computational biology, microbial ecology, molecular biology, microbiology, soil biochemistry. And all these people, working in teams have to work together towards a common goal. So now in the field of soil microbial ecology, we cannot work alone anymore. We need to work within interdisciplinary teams. And it's not always easy. There are best microbial associate and best plant also. So we could think of selecting both plants and microorganisms to form an association between plants and microorganisms that are going to be particularly efficient at using nutrients. And Well, I'm not very good with plant genetic selection, but I know that you need to phenotype your genotypes, and this can be done, for example, by measuring yield. But the problem, it's not a big problem, but what happens is when you have the yield, you don't have the plant anymore. However, there's more and more technology that is developed to make the selection of plant faster. And some of the way is using remote sensing. Remote sensing is interesting because it gives you information on the plant when they are in the field. And when the plant is in the field, and it is particularly effective, for example, at using nutrient, then we can go and look in the root what kind of microorganisms are there. And maybe this genotype of plant is successful because it has very effective metabolism in the leaf, but maybe because it has very successful combination of microorganisms in the root. So before I finish, I would just like to point out a few obstacles that are slowing down the process of development of application, microbial application for improving nutrient efficiency in crop. And one is the limited communication between disciplines. You know, I'm a microbiologist. I don't read the literature in other fields. We also have different culture. For example, in agronomy, people are very serious with statistics, but in other fields, maybe people don't do statistics at all. So we can have problem understanding each other there. There's a problem of security, of course, the problem of administrative rule and processes that can be maybe included in the security aspect too. We need certification, for example, so this is very complicated. But there's also a problem when different research scientists of different institutions try to work together. There's also a lot of agreement and paperwork and restriction that are going to apply. Research funding structure also needs to be changed, because if we work with big interdisciplinary team, then we need to form this team. The people in the team need to learn how to work efficiently together. And we also have to have all these agreements signed. So if we have a 3-year grant for doing something, we are not going to do much in a big interdisciplinary team. Fortunately, more and more, at least in Canada, we have these 5-year grants that are very helpful and help us getting significant results. So I've tried to show that we need to use nutrients more efficiently in crop production. Soil microbial resource offer some possibility towards this goal, and developing biotechnology is quite challenging these days. So thank you very much for your attention.